welcome to our event on de-extinction. I am Ann Merchant and I work with the National Academy of Sciences. And of course, I am joined by...
I'm Rick Lovard, Program Director of the Science and Entertainment Exchange program of the National Academy of Sciences. And I must say, Ann, you gave us just enough time to read each slide. I thought you did a great job.
Thank you very much. And of course, you do a great job putting together those decks. So it's a team effort, and I appreciate that. So thank you. And of course, we appreciate all of you being here today. We have another great event, at least I think so. And we're very glad that you're here for us today. We usually start by giving a little context for the event. I like to sort of point out some of the work of the Academy that feels relevant. And interestingly enough, when I went into the National Academy's website and started typing in things like de-extinction, I got nothing. We have not done a lot of work on this topic. Of course, some of the underlying science that is relevant to de-extinction on genomics and on Crispr and things like that, we do have a fair amount of work in that area. But of course, it's not relevant specifically to the talks that we're going to hear today. But if you are interested in some of that underlying science, please do go to the National Academy's Press and look at some of the reports. They're all there. But of course, we do recognize that a lot of those big reports, that the audience for those reports are very specifically, as we like to say, decision makers and policymakers. And we know that some of those people may be in our audience today, and you may be familiar with those reports. But the reason that the Science and Entertainment Exchange exists is because, well, not everyone wants to read those reports. And so, Rick, perhaps you'd like to say a few words about the exchange.
Sure. So if you are a writer, producer, director, studio executive, storyteller, really in any capacity, you have a question about science, you can call us and we will connect you to an expert. We've done this over 3,500 times, including many, many, many Hollywood summer popcorn movies that you may have seen. I think we've worked on every Marvel film since Iron Man 2. Just to name a few. We also work on documentaries, graphic novels, and video games. If you are a storyteller, please give us a try. If you are a STEM professional and this is your first time becoming aware of the work we do, we are always looking for volunteers to connect to storytellers. So please do put up your hand. And I think Sachi's putting some info down there for you to contact us. So you can find a recording of this show on the Exchange's website starting, I think, on Friday. It should be up. Usually it is, or you can subscribe to the Science and Entertainment Exchange on YouTube to see these events and other videos like it, or our Ask a Scientist series. You can sign up also for our weekly newsletter, Collider. So when this event is available, by the way, if you are watching it live, you will get an email and we'll say this event is available. So there you go, click the link. I want to thank our sponsors today, Howard Hughes Medical Institute. And we also get major funding from Alfred P. Sloan Foundation, as well as so many individual donors like you. I know there was a slide at the open deck. I want to thank everybody who donated to this event, specifically, especially those who joined at the supporter level. We really appreciate it, and it goes right back into the work we do here. So today, we have two amazing speakers. I can't wait to hear from them. Ann will tell you more about them in a second. They will each have a few minutes to speak generally about the topic. And then Emily Carmichael, the writer from Jurassic World Dominion, among many other big Hollywood blockbuster movies, will come on and she'll be in conversation with them. If at any point during any of that, you have a question, you can put it right down here in the Q&A. Some of you have it up here, but where it says Q&A, you can put that in there, and then I will be dutifully giving your questions to Emily. If we don't get to your questions, so sorry, that's on me, that's not on Emily. We get to as many as we possibly can every week. And my rabbit hole item for the week, which is that thing that as we were researching this event, I just couldn't stop reading and had to get to the end of the article, was why so many bugs are going extinct. There were a bunch of articles on this topic last week, but this one in particular caught my attention. And spoiler alert, it's our fault, humans. We kind of stink. Anyway, back to you, Ann.
It's also a little bit of a spoiler, well, maybe a tease for our next event, which is, of course, on insects, not on their extinction, but on their barbarous and weird ways. So that's a little bit of a tease for our next event. And my rabbit hole was really in some ways, the inspiration for this event, which was the 2013 TEDx event at the National Geographic Society on de-extinction. Sachi is putting a link in the chat window there. Not all of those videos are up on the TED website, but some of them are, and there are some other de-extinction talks that that you might want to check out. That was a really interesting event. It's what introduced me to the topic, and it's what introduced me to both Beth and to Hank. And I thought, you know, not only is this an interesting topic, but these are two amazing people to introduce all of you to the topic. On the one hand, we have Beth, who will actually talk about the science. I think Beth is probably going to tell us that we probably should not expect to see Jurassic Park dinosaurs in a theme park near any of us anytime soon, or the woolly mammoth necessarily grazing in a field near us. But she will give us some good grounding on the science. And I was so impressed by the way that she presented at that National Geographic event. We've had her before on an exchange stage. And the great thing about Hank is that he, I believe, was the last speaker at that event where you'd heard a whole bunch of people for a full day talking about de-extinction. And he really pulled it together for us to make us understand why this was the best idea in the world and the worst idea in the world, leaving most of us, including me, thoroughly confused. I don't know if that's how we'll leave you, but Emily will help us get to the bottom of it. So I'm going to go away, as will Rick, and we're going to hand this over to Beth. And that's what I know you've all been waiting for. So Beth, please turn on your camera while Rick and I turn off ours, and we will let you take us from here.
All right. Well, that was quite an introduction and also a spoiler. Not cool, I'd like to say, to start off there. Like, I'm just going to say, no, we can't do it. And that's going to be it. Why are we even bothering? So I'd like to start by saying, no, we can't do it. And that's going to be it. No. So as has been set up here, we've divided this into two parts for the intro. I am first going to talk about the can we aspect of de-extinction, and then Hank will join us to talk about the should we aspect of de-extinction, and then we will be grilled by the amazing Emily, and I'm very excited about this. But okay, so how do I set this up? Mostly when people think about bringing extinct species back to life, the thing that immediately comes to mind is the idea of cloning. The problem with cloning is that this is a very specific scientific process that's actually known as somatic cell nuclear transfer. The idea is that our bodies are made up basically of two types of cells: somatic cells, which are skin cells and heart cells and liver cells and brain cells, etc., and germ cells, which are sperm and eggs. Normally what happens is a sperm carrying one copy of the genome and an egg carrying another copy of the genome fuse to create a zygote that will eventually start to divide and differentiate and become all of the different types of somatic cells that make up a whole organism. In cloning, what you want to happen is that something that is already a somatic cell forgets all of the instructions necessary to be the type of cell that it is and reverts to that early form, so that it, which already has two copies of the genome, which we would normally get one from mom and one from dad, instead, this already has two copies of the genome, can start to divide and differentiate and become a whole organism, just like a zygote would. The way that we do this is we start with an egg cell from something that is alive, and we suck out the nuclear material. So now it has 0 copies of the genome. And then we take a somatic cell from somewhere. In the case of the most famous clone, Dolly, this was a mammary cell. And we stress it out, starve it of nutrients so it's stressed out. Stick these two things next to each other, zap them with a bit of electricity. The surface of the cells opens up, and that nucleus, which contains both copies of the genome that we would normally get, one from mom and one from dad, and they dump into the egg cell. You zap it again, and then it does the magical reset, and it starts to become everything that's there. This is great. We know how to do it in lots of different mammals. But the crucial thing for de-extinction is that we have to start with a living somatic cell. For anything that has been extinct for some time, anything that doesn't have a frozen tissue, especially frozen in a very particular way that it was taken while it was alive and stuck into the deep freeze so that it stays alive, we cannot do this. There is no possibility of cloning something if you don't have a living cell to start with. So what will we do instead? Instead, we would take a living cell from something that's close evolutionarily to the thing we want to clone. In the case of a mammoth, we often think of an Asian elephant. They're about 6 million years diverged from each other. It's about 1.5% of their genomes. The genome is 4 billion letters long. So 1.5% of 4 billion is a lot of evolutionary differences, but we take that living elephant cell, and then we can use the tools of gene editing, genome engineering, to gradually cut and paste our way from an Asian elephant genome in that living cell to a mammoth genome. How do we know what the mammoth genome looks like? That's something we can do. We go out in the field, in my field in ancient DNA, we collect really beautifully preserved mammoth bones, we can extract DNA, it's in terrible shape, it doesn't matter. We can line up those tiny little extracted broken bits of gunged up DNA pieces on a computer against that elephant genome, and gradually, using that computer, figure out what all the differences between these two species are. And then we can make those changes in that living cell, in a dish, in a lab. Can we make all of them? No, but we can probably make some of them right now. Maybe that technology will improve. And so we can focus on things like, if we want to transform an Asian elephant into a mammoth, genes that maybe allow it to survive in a really cold climate or to eat the kind of stuff that a mammoth used to eat. And we can tweak those cells until we have that. And then we have a cell that's alive, that's growing in a dish in a lab. That's great. That's step one to cloning. Next, we have to transform that into something that's alive. So we get an egg cell from something, we suck out the nuclear material, we inject this into there, we start it dividing and differentiating. And then if all that goes well, let's just say if all that goes well, then we have a little developing, tiny little early stage embryo in a dish in a lab. Then we need a mom. Who's the mom going to be? Well, again, you need something that's evolutionarily close so that the pregnancy might actually work. We need to be able to know how to move that thing, that developing organism from a dish into that mom. We don't really know how to do that with elephants yet. We have to make sure that all of the signaling for development goes right. This is tricky because during development in utero, a lot of that signaling comes from the mom's genome, not from the baby's genome. Remember that mom is not the same species as what's there. And then let's assume all that goes well. It's born and it has to somehow learn how to become that animal. How do we do that if we don't have that animal anymore? Lots of potential problems there. And also, this whole process really only works for things that we can clone. So for now, things like birds and insects and amphibians and fishes are out of the question. We need new technologies for that. We can do mammals, maybe, and maybe also marsupials. So that's pretty much where we can get with can we, the very specific technicalities. I'd be happy to answer any other questions, but in the interest of leaving time for talking about the cool and fun trickiness in there, I am now going to pass it off to my friend and colleague, Hank Greely, who will talk to us a little bit more about whether we should.
Well, thank you, Beth. And I want to thank Anne for that introduction, tagging me as somebody who is terminally confusing. I'm afraid some of my students would probably agree with you, but I'll try to be less confusing than usual here. I want to point out first something important, leading on from what Beth has said. Except for cloning, if you're going to try to gene edit an existing species genome in order to bring it closer to an ancient species genome, at least for the next few decades, you're not going to get an exact genetic copy of that extinct species. There are, say, a couple million differences between mammoths and Asian elephants, and we're not able to make a couple million edits in an Asian elephant's genome. So what you'll get is kind of a genetic hybrid. It's a bit of Asian elephant, it's a bit of mammoth. You can call it an Asian elephant with mammoth characteristics. Some people call them mammothants, but it won't be identical. Now, in 20 years, maybe we will be able to make millions of changes. Maybe we'll be able to grow to build the mammoth genome from the starting blocks and make it completely, exactly identical to an ancient mammoth's genome. It still won't really be the same species in some respects. It's not going to have the same environment. For one thing, there'll be 50% more CO2 in the atmosphere. It's not going to have the same microbiome. And I think, interestingly, it's not going to have the same culture. I am quite confident that mama mammoths taught baby mammoths something about how to be a mammoth, that mama Asian elephants are not going to be able to teach these hairy little calves that they have. So we're making, at best, sort of facsimiles of extinct species, but not the species itself. Having said that, let me talk a little bit about some of the pluses and minuses of the possibilities of de-extinction. I do want to note that although my work is in the ethical, legal, and social issues raised by the biosciences, I love what I do. I think it's kind of important. The events of the last 10 weeks in Ukraine are kind of a reminder that there are different levels of things of ethical importance. And what we're talking about today, although important, pales in significance to other ethical issues that are ongoing in the world and in our society, but they're still interesting to talk about. Let me start with some of the downsides, potential downsides of de-extinction. To me, the most important is the issues with respect to animal welfare. What happens if trying to make baby mammoths hurts the Asian elephant mothers who are carrying them? What happens if half of those baby mammoth calves or mammothant calves end up seriously disabled or suffering? The possibility of animal suffering is a real concern, at least if you're de-extincting animals. If you're de-extincting plants or fungi or microbes, we're not very worried. But for de-extincting animals, I think there are good reasons to be concerned about animal welfare. There's a second concern that if they got loose in the wild, these things could wreak environmental havoc. They could be like the kudzu taking over the American Southeast. I'm not too worried about that for the most part. Our ancestors were pretty good at killing mammoths with spears. If we needed to round up runaway mammoths, I don't think we'd have that much trouble. If on the other hand, we were trying to round up runaway passenger pigeons, that might be more difficult. And if we were trying to round up runaway microbes, if somebody were to sort of de-extinct the kind of extinct smallpox virus, we could have real trouble putting that back into the bottle. There's a third set of issues, and to me, these are in some respects the most important, the sort of political issues. Will the possibility of de-extinction do one of two bad things? Will it dry up and steal funding from important conservation biology efforts to preserve existing endangered species? That's something a lot of people working in those efforts who have far too few resources as it is are very worried about. Or will it give people who aren't very worried about endangered species an excuse to say, oh, let's go ahead and build that factory. Think of all the jobs that factory will provide. And if it drives a snail darter extinct, well, we can always bring it back. Sort of the moral hazard argument. I think these are serious arguments. They're not necessarily completely unanswerable arguments, but they're things that are to be worried about. The last argument is the one that I think is most viscerally powerful, although frankly, it's the one that bothers me least. It's that it's unnatural. It's wrong, it's against God's will, it's against Darwin's will, it's against evolution's will. People latch onto that. My own view is that pretty much everything humans do and have done for the last 100,000 years is unnatural, including the unnatural way in which we caused many of these extinctions. But I do think that that sort of naturalness argument has a lot of visceral appeal to people and would apply to some de-extinction. Now, that's a pretty heavy set of concerns. What's on the plus side? Well, there's some scientific benefits of being able to see even a reasonable facsimile of a mammoth. We could learn a lot about mammoths, even though it's not quite a real mammoth. There are some possibilities for driving technology, and I love that Beth mentioned the problems of trying to use these technologies in birds. People are working now on trying to solve these problems with respect to genetic manipulation of birds in ways that could have carryover effects for all sorts of bird conservation efforts for endangered birds, of which there are hundreds, or improvements in existing domestic birds or wild birds, make them disease intolerant, et cetera, disease immune, et cetera. So I think there's some technological advantages. I think there's another set of advantages that I think has to do with, well, the noble way to phrase it is awe. I still distinctly remember the first time I saw Yosemite Valley. I was 14 years old. We went through the tunnel, rounded the corner. There was the valley, and that view changed my life. That sense of awe. I think people may get that sense of awe from seeing a mammoth or a saber-toothed cat or even a passenger pigeon. And I think that's a good thing. Now, the less noble way of explaining it, which is more frivolous, but maybe more human, is it would be cool. I think we underestimate cool as an advantage. Much of what we do in our lives, like tourism, for example, is to do things that are cool. And I think it would be seriously cool to see a giant ground sloth. That would be on my bucket list, though I don't think it's likely to happen during my lifetime. There is a final possible advantage that I put in kind of the speculative camp, and that's the possibility that reintroducing some of these species could help the environment. There's an argument that reintroducing mammoths into the Arctic could help the Arctic environment. It could even slow global warming. I think that's an interesting possibility and something to be studied. I'm not going to put a lot of money on it myself. So there's some pluses, there's some minuses. I hope you're not terminally confused, but a little confused. I'm okay with that. Thanks. And let me turn things back over to our moderator.
What's up? I'm here to moderate this conversation. Thank you so much, Beth and Hank. It was so interesting to hear from you guys. And first of all, what I want to ask is, do you have questions for each other after hearing those presentations? I know you guys are familiar with the work of the other, but did anything come to mind that you maybe want to start us off with?
Hank and I have done these events so much together that I'd be afraid if I asked him something that wasn't relevant to this particular conversation.
So I do have one for you though, Beth. I'm curious, how big a barrier do you think it's going to be to the technical barrier with non-mammals?
It's hard to know. I think if you'd asked me that question in 2013, when we first did this TEDx, I would have said that the barrier was pretty high, but there's a nonprofit that's out of Sausalito called Revive and Restore that's recently been reaching as deep in as they possibly can into the world of scientific knowledge about bird reproduction and engineering. And they've put out a call recently to try to bring people together, new minds, different ideas, hard, same problem. And I think that given this attention and investment into trying to solve this problem for birds, we're a lot closer today than we would have been before. There's at least several strategies that people are considering, which is way better than just thinking about one strategy and how we're going to do it. The problem with birds is that you can't get access to the egg cell at that stage where it's ripe and ready to be fertilized, but not yet fertilized. By the time a chicken egg is laid, for example, it's a 24-hour-old embryo. And at that point, you can't suck it out of the egg because it's a lot of different types of cells and it's doing a whole thing. It's like almost ready to be a bird, right? But what you can do at that point is you can stick a needle into that egg and you can suck out the primordial germ cells, the cells that will eventually someday become either sperm or eggs, depending on the biological sex of the organism developing in there. And you can edit those, and then you can re-inject those into an egg cell at the same developmental stage, and they will go around the outside of the body and become part of the gonads of that organism. But then you've only edited one of the copies of the chromosomes that you need. Remember, we have two copies, one from mom and one from dad, and together that makes our genome. And we really want to have edited both of them. Of course, you could then fertilize it with edited sperm, and then you would have these edits, but it's then twice as hard to make the right edits and to make sure those edits go in. So this is one of the strategies people have been considering for birds. It might sound crazy, right, that you can do this, but we know that this works because people have injected chicken embryo eggs with duck primordial germ cells. And then those duck primordial germ cells swim around the outside of the chicken, lodge themselves in the gonads. And if you fertilize that chicken, then after she is born and she's a perfectly normal chicken with duck sperm, those duck sperm will only actually fertilize the duck eggs. And that chicken will lay a bunch of beautiful, wonderful, normal eggs that will hatch into perfectly normal ducks. Right?
If it quacks like a duck, it's a duck.
Wow, that's phenomenal. So Beth, do you think the de-extinction of bird species, just because we're on that topic now, do you think that's inevitable? It's clearly there are a lot of challenges, but do you think that's something that's going to happen in the next 150 years, even if it's not going to happen in the next 5 to 10?
Well, this is something that we have to say sooner or later at some point, then it depends on what you mean by de-extinction, right? If you are willing to accept something that is a bird that contains some traits of an extinct species, then I think, yeah, we will get to the point where we can do that. And I think that's great, because that means we have technologies that we can use to edit birds that are alive today and not yet extinct, but perhaps in danger of becoming so, and will give us all sorts of new and exciting powers for conservation of biodiversity. If, though, you are asking whether we will ever get something back that is 100% identical genetically, behaviorally, physiologically to something that is extinct, then I think the answer is no. We're not going to be able to do that. And that is not just because of this technological problem, but because of the truth that everything that is alive is more than the sequence of A's and C's and G's and T's, the letters of the DNA that make up our genome, that can make up our genome, right? We are different. There's something more to us. If all it took was the A's and C's and G's and T's in our DNA, then identical twins who are genetic clones would be just replaceable as humans. You could just swap one out for the other one. And we know that that's not true, right? Who we are and what we are depends on the environment that we're exposed to, both before we're born and after we're born, and the DNA that is in our cells. So even if we can reconstruct that DNA, can we then reconstruct everything about an environment that's extinct in order to recreate that? I don't think we can.
And that environment is always changing. And in a way, it means that humans today aren't the same species as humans 20,000 years ago or 10,000 years ago, let alone 100,000 years ago, because the environment changes us. And pigeons today aren't the same as pigeons 1,000 years ago. It's always a moving target. You never step in the same species twice.
Speaking of those changes, you mentioned briefly, talking about like microbe resistance and stuff like that. Can you tell us a little more about microbe resistance? Like, is it the case that an animal, let's say we're talking about that scenario of a relatively detailed reproduction of ancient DNA, even though it's not going to be identical to that ancient DNA. If we were able to bring that animal back, is it the case that it would be instantly laid low because it would be incapable of handling our microbes?
That's an interesting question. Well, I mean, we have experience with humans in this way. We know that when Europeans first arrived in the Americas, that they carried with them diseases to which people were not exposed to, the indigenous people were not exposed to, and it caused horrible things to happen. And we also have experience, this is a kind of a scientific way of thinking about this, from the Neanderthal genome. When we compare the Neanderthal genome to human genome, it allows us to better understand what genes moved between Neanderthals and people when Neanderthals and people were interbreeding with each other. And we know that some of the genes that have moved from Neanderthals into anatomically modern humans and then gone to really high frequency, higher than is expected by chance, which means there must have been some pressure on that population for these genes. They must have done some good for the population that got them, are MHC genes. These are genes associated with resistance to disease. The idea there being that people, anatomically modern people, moved into a place where Neanderthals and their diseases had lived, and having those Neanderthal evolved genes in defense of those diseases provided an advantage to the people who were there. So it is possible that if one were to be able to very cleanly reconstruct a thing that lived a long time ago, then maybe it would be immediately exposed to diseases that were here. I have to say, though, the MHC region of the genome is super difficult to actually reconstruct. So it will be one of the last things that we can possibly fix to look like an ancient genome. And also,
tell us more about the MAC. This sounds like exciting and cool.
It's not exciting and cool. It's just very diverse and divergent with lots of different repeat motifs. It's hard to figure out how things go. And it's because there's a lot of diversity there. And that's, because it's evolutionarily useful for there to be lots of diversity in different people or different organisms in this part of their genome, which is responsible for scrambling up different ways to be resistant to diseases.
We see the same thing in non-humans as well. When plants and animals move across continents, they're exposed to different pathogens. Sometimes those pathogens are so bad that they can't take root in the new place. Sometimes, most of the time they do. So No, I suspect extinct animals would have a higher disease burden early on if they were brought back, but some of them would survive, and their major histocompatibility complex genes would evolve in ways that protect them.
Yeah, that's
Now, is this when
MHC. Sorry
we're
about that.
talking about, can you guys just define epigenetics for us? We had a question touching on the topic of epigenetics, and I know that's a word that people are hearing a lot, and I think they would appreciate a succinct scientific definition.
That's for you, Beth, not for me.
There is not a succinct scientific definition. In fact, I read a really good Twitter thread about this just the other day, and I wish I could remember who tweeted it, but it was somebody who was saying, you know, epigenetics means at least 3 and possibly more different things to scientists. And when people just say epigenetics, what they really mean is stuff that's not actually encoded in the DNA, but it actually means lots of different things. And they all get kind of, and even among scientists, people confuse all these different definitions. I think most commonly people think of it as something that is inherited, but maybe between generations. You know, I saw the question had to do with marks in the DNA, and that people sometimes think of as marks that have to do with when genes are turned on and when genes are turned off. And these are, of course, different in different cells. You know, you don't need the same genes to be turned on to be a heart cell as you do to be a brain cell or a skin cell. And so some of these marks emerge during development so that they can tell different cell types what genes to turn off and what genes turn off at different extents. Sometimes these are a response to environmental influences. So the environment is different in one place or another. Your proteins need to be produced in a different way in order to be able to make you thrive and when it's really hot versus when it's really cold. And there's also a bunch of it that we don't really understand. I mean, sometimes epigenetics is just used to refer to stuff we don't quite get as geneticists, right? So there's not a succinct definition of what it means. I saw that the question was, does it matter that we don't know what the epigenetics are? I don't know the answer to that, right? There's a lot of things that we don't know. And does it matter? It depends, again, on what you are willing to accept as something that's de-extinct. If you are willing to accept a slightly hairy Asian elephant that can survive in the cold as a de-extinct mammoth, then I doubt some of this confusing, vague, arm-wavy epigenetic stuff is going to be important at all. But if you want something that is 100% exactly like a particular mammoth that used to be alive 30,000 years ago, then yeah, probably would need to know what epigenetic marks were on that particular organism's genome. Is it important? Depends why you want to do this, right? Yeah. I would say no.
Same thing's true, I think, for the microbiome. I mean, we have more non-human cells in us, about as many non-human cells in us as we have human cells. And those undoubtedly have changed.
I thought it was like a ratio of like billions to 1. I thought there were like billions of non-human cells for every one of your cells.
No, it's about 1 to 1.
It's about 1 to 1? Yeah. That's so much less dramatic.
Sorry. And they only weigh about 3 pounds. It's about 1.5 liters of non-human cells because they're tiny, but they're important. And, you know, the mammoth probably had different ones than the current Asian elephants do. Will it be a big deal? Who knows? Probably
Fun fact,
not.
Though, about elephants, when a baby elephant is born, his mom feeds him a little bit of her own poop to help to establish that community of microorganisms that they need in their gut to digest their food that they eat. So this baby, slightly edited elephant to be more cold adapted mammoth-like thing will certainly have an elephant's gut microbiome to start with.
You've really changed my view of elephant motherhood in a way that it may be TMI.
Oh, it's a beautiful thing, Hank.
Don't tell my kids.
We have a question, which is about CRISPR. Can you tell us a little more about CRISPR? Because that's another thing that folks have heard about a lot. It looks like somebody has the question, which is, has CRISPR or any technology that might be used for de-extinction had a positive impact or increase purely because of the pursuit of bringing extinct creatures back to life? Or does it all piggyback on existing technology? I think what that question is how much has this enthusiasm for the possibility of de-extinction energized CRISPR and other forms of genetic research? And are there benefits that we could get from that?
I think that a nice way to answer that question would be for Hank, who's talked a lot about CRISPR, to talk a little bit about what CRISPR sort of allows. I don't think that the enthusiasm for CRISPR comes from de-extinction. I think the enthusiasm for CRISPR is driven mainly by the potential of this technology to improve human health. But there are a lot of ethical conundra, is that the plural for conundrum, around this? So
issues.
It's okay. Hank.
Easier to spell. Yes, so I don't think de-extinction is driving CRISPR. I think CRISPR is helping to drive de-extinction. CRISPR is another one of those great acronyms like MHC. It's clustered regularly interspaced short palindromic repeats. And I think we all need to be deeply grateful to the Spanish and Dutch scientists who came up with something that led to a nicely pronounceable acronym like CRISPR. It's just such a good acronym because you can turn it into a verb and it fills all parts of speech, right?
Unlike de-extinction, which you cannot conjugate in a useful way.
Right, but they're CRISPR babies, you're CRISPRing something, it's a great word. And it is a really important breakthrough, big way to make genetic changes, to make genetic editing. We could do it before CRISPR. I like to say CRISPR is the Model T of genome editing. There were cars before Model Ts, but they were really expensive and they broke down all the time. And only rich people could have them, because you needed to have a full-time chauffeur/mechanic to make them work. And then Henry Ford came along with the Model T, and anybody could have a car. And pretty soon everybody did have a car, at least here in California. And that's what changed the world. It wasn't the first car, it was the first easy to use, easy to buy, cheap car. And that's CRISPR. So CRISPR is making a lot of things possible. I remember the first de-extinction meeting I went to was in February of 2012 in George Church's lab. And he had this complicated system that he called MAGE that was going to edit a bunch of stuff. CRISPR comes along the next year and later that year, actually, and MAGE kind of disappeared into the junkyard of history because CRISPR was so much better. And it's not perfect.
Of course, in my field, your friend would have then gone on to edit his own genome with MAGE in order to prove its superiority, would have had a horrible accident and become a terrible villain.
Well, we are talking about one of the more interesting scientists around in terms of in George Church. So I think there are movies to be made about him.
There's a room in his lab that he can't go into because there are loads of his own cells that have been transformed in crazy mutagenic ways. And he's worried. The idea is that he can't go into this room, not because he would infect these cells, but because these cells could get into his body. And they're so similar to him that his own body wouldn't know that they were cancer or mutagens or crazy and would just accept them as their own, and it would be terrible. So there's a good movie.
Yeah, there's a good plot.
A lot of good premises here.
Personalized George Church cancer. That's not what people usually mean by personalized medicine through genetics, but it is a good plot possibility.
I'm going to keep taking these audience questions. These are good questions. Somebody asks, how are Indigenous peoples being consulted around these kinds of research? And are there other specific benefits and challenges from de-extinction highlighted from these conversations?
I think the consultation of Indigenous people depends on what projects you're talking about. There actually aren't that many active de-extinction projects. I mean, considering how much media attention the idea gets. There's the Mammoth Project. I saw there was a question about who's actually trying to do it. There are 3 different groups that at the moment that are very interested in mammoth de-extinction. There's the Japanese team, a South Korean team, and then a team that was led by George Church. He's recently started a new company called Colossal with tech entrepreneur Ben Lamb, and Colossal are now working quite diligently at the process of creating a mammoth. The Japanese team and the South Korean team are both focused on cloning, which, as I've said before, no living cells, that part's not going to work. George and Ben's team at Colossal are really thinking about using gene editing technologies to do this. There is a challenge of consultation with Indigenous people because mammoths actually lived everywhere across the Northern Hemisphere. So one would have to consult with everyone that used to be alive. Of course, not everyone would have interacted with a mammoth by the time they went extinct. But I think that it is important to think about who who are the stakeholders in any one of these projects. And in this case, the stakeholders for mammoth de-extinction would depend on what the outcome was. Right now, they're working with Sergei Zimov, who has this place up in northeastern Siberia called Pleistocene Park, where he would like these mammoths to go, and also talking to people at different zoos who might be interested in having these animals there. There are projects, though. The Tasmanian Tiger Project is working very closely in consultation with Indigenous groups in Australia about whether it would be nice to have Tasmanian tigers back, where they might go, how they might be helpful in relearning projects and things like that. So I don't know, Hank, did you have any other?
Just that this has been a bad aspect of a lot of genetic research and archaeological research and anthropological research for the last couple hundred years. And there are good signs that it's moving in the right direction. It isn't all the way there, but geneticists working on Indigenous people's histories and backgrounds are increasingly consulting with, including in the decision-making, including in from the very beginning, Indigenous peoples. There's still a ways to go with that, but that's an important trend. And to the extent the de-extinction overlaps with Indigenous issues, that Indigenous peoples are stakeholders in it, they certainly should be included in the discussions.
Yeah.
What was the second half of that question? I forgot.
Just, are there other specific benefits and challenges from de-extinction highlighted from those conversations? Just, if there are ongoing and vital conversations with Indigenous people about this area of research, has it brought anything interesting or important to light, as one would expect that it might? But it sounds like we don't have right now a very strong highway of access to those ongoing conversations. And it sounds like we think those ongoing conversations are probably fairly lacking.
Yeah.
On that topic, who are the people who are making these types of ethical decisions? Are you guys, does each of these gene projects have an on-staff ethicist? Hank, are you being consulted about stuff like this? Do you feel like we need some sort of regulatory body to be answering these questions? Whose decision should it be?
So it should be our decision, meaning people's decisions. I'm a Democrat, certainly with a small d. I think ultimately these are political decisions that need to be made by the voice of the people as reflected through their institutions. I don't think anything is imminent enough that we need legislation on this. I do think, though, that this is a subset of a bigger issue of regulating CRISPR use in non-humans, whether it's for agricultural purposes, livestock purposes, public health purposes, like efforts to end malaria by modifying mosquitoes. Mm-hmm. Those do need some significant regulatory changes and improvements that I think do call for legislatures, for governments around the world to get together and try to figure things out in consultation with, taking advice from scientists like Beth and ethicists, and law and bioscience people like me and others. But ultimately, it's not our decision. It's not the ethicists, we're not the priests who send you to heaven or hell. And the scientists aren't the philosopher kings who make all the decisions. Some of them, I think, occasionally think they should be. Yep. Some of the ethicists occasionally think they should have the keys to the kingdom, but we don't. We advise, we consult, we suggest, but ultimately governments, people working through governments and working through their own individual decisions need to decide.
Yeah. So I was going to ask, we have a question here, which I think is very important, which I want to get to in a second. Beth, I was going to ask if we couldn't fulfill the Jurassic Park premise of bringing dinosaurs back to life from their blood encased in a mosquito, which it sounds like the fundamental problem is that those are not living cells, and we need living cells in order to be able to use cloning technology to bring dinosaurs back to life. My follow-up question was going to be, well, what about reproducing the mosquito? But it sounds like, once again, we are going to have the same problem of not having living mosquito cells. Is that it in essence, is that we need living cells and we don't have them? Is that why Jurassic Park cannot happen?
Well, we need living cells for cloning, right? Mm-hmm. If one were to be able to reconstruct a non-avian dinosaur genome and also then identify an appropriate surrogate maternal host for this,
Surrogate
then
maternal
you would
host.
be at exactly the same place as you are with mammoths and other things. The challenge, a fundamental challenge with non-avian dinosaurs is that there is no DNA remaining in any of these remains, right?
Why not?
The oldest DNA that's ever been recovered is probably a little bit over 1 million years old. This was published last January. It was a mammoth bone. And that bone was found in permafrost, which is dirt, sediment that is frozen and has been frozen since the time of deposition. So once an organism dies, the DNA that's in its cells starts to be broken down, chopped up into tinier and tinier pieces. There are no living cells, and that's not the only problem. The other problem is things happen, like UV radiation from the sun hits the DNA and breaks it. It does that during life too. It's why we wear sunscreen when we go outside. We, of course, have proofreading enzymes and repair enzymes that can go through and fix the damage caused by UV. Otherwise, every time we went outside, we would get cancer, right? Wow. But this proofreading and repair is an energy requiring process. And after you're dead, there's no more energy. So these types of damage just accumulate in the DNA. Also, when there's enzymes in our body, there's enzymes in microbes and fungi, they get into the cells and chew up the DNA. And in hot places, this can happen really fast. DNA might not survive for more than a couple days or a couple weeks, but in really cold places or where it's dark, there's no UV, then the process is slowed down. It's not stopped entirely, but it's slowed down, which is why much of the early work in ancient DNA research focused on things that only lived in the Arctic and really cold places. That's why we have so much information about mammoths and things like that, and not very much information about giant ground sloths and things that lived in warmer places. Mm-hmm. So 1 million years old, dinosaurs went extinct more than 65 million years ago.
Wow. So what you're saying is we need time travel.
Yes.
Right.
Time
so
travel.
Next fall,
one of the many things we need to bring dinosaurs back.
To make this happen.
But let me ask you a question, because we've got to answer. What you've thought about this a lot. I mean, in your mind and your imagination, which is amazing. What would you want back? I mean, if you got to choose, what would you want us to have?
I mean, look, like the vision of I think the Jurassic franchise has always had this idea that maybe the dinosaurs could have a sanctuary. Maybe there could be a place where they could live in harmony with the environment of today, where they could be animals, where they could enjoy animal behavior. And maybe, just maybe, we could look at them every once in a while. I do have to say, I think that would be pretty great. We had some questions that were about like why mammoths in particular, and I think that's a great question. Somebody asks, the world is undergoing a huge mass extinction of insects, which is underreported because insects are so essential for our survival. Is there any work ongoing with insects? Why have mammoths been elected as the flagship animal for these experiments?
Yeah, I don't know the answer to this. I always answer this question the same way. I say, as soon as people realize that we can't do dinosaurs, they automatically just turn to mammoths. Don't know why. I think it's just because they were big, wow. We know that they've been gone for a long time. They're really iconic of extinction. It's sometimes the dodo, but often the mammoth. Yeah.
But like nobody makes animated kids movies about extinct insects, but the Ice Age franchise is up to, I don't know, 4 or 5 movies.
Wow.
We like these Pleistocene megafauna, probably because we saw them and they're warm and fuzzy, and we like dinosaurs because we all learned about it. There's some really cool, huge mammals from 50 million years ago that would be really interesting, but nobody knows about them, and some huge amphibious things from 250 million years ago. But for whatever reason, they didn't get into the culture the way that dinosaurs, and the fancy term for it is the Pleistocene megafauna, but basically the Ice Age big stuff.
Megafauna
got into the culture.
So wow, so this the focus on this particular species and the use of mammoths as the testing ground and as the bar to be achieved is more for cultural reasons than for anything scientifically special about that species.
In fact, if you wanted to do a test case, the mammoth might be one of the last things you would try.
It's terrible.
You're dealing with an endangered species of Asian elephants.
Right.
They are big. We don't know how to do elephant IVF. We don't know.
They don't reproduce in the wild until they're 14 or 15 years old.
It takes them 22 months to do a pregnancy. I mean, imagine being pregnant for 22 months.
Wow.
You'd really like to do like a mouse.
Yeah, something
Yeah.
we know a lot about, or a fruit fly, right? We know a lot
Right,
about fruit
but
flies.
de-extincting a mouse is probably not going to get you a lot of attention, money, and interest.
But I should say, I think one of the reasons that I am excited, even about all of the progress that's been made, just pushing toward mammoths, and one of the bits of pushback that we often get working in this is that the de-extinction, this idea of de-extinction, is taking money away from conservation work that we know has higher efficacy, that actually can achieve something in a reasonable time frame. And we've always pushed back against that and said, we're not saying de-extinction instead of conservation. We're saying that we need this technology in addition to conservation. And I really strongly believe that any of the effort that goes into figuring out how to do this with a mammoth or with a dodo or with any other extinct species is going to find an easier path toward doing something useful with species that are still alive. This is technology that we sorely need, that is going to be applicable much more straightforwardly to conserving and protecting species that are alive today. And also when we see this investment in companies like Colossal, which just recently went through a Series A and raised something like $75 million, right? This is new money to conservation. This is not the same old people donating money to technology that might be useful for conservation. It's new money for new tools. And this is exactly what we need in the time period where we do have a mass extinction event underway. And we need as many tools from science and science fiction as we could possibly get in order to try to stop this hemorrhaging of species that's going on today.
And as many good stories as possible to bring the money in to make and use those tools.
Yeah.
Wow. Wow. I know a little something about that. So somebody asked, what is the feasibility of avoiding inbreeding problems in early generations that cause problems later? So once you have a representative of the species that you're trying to de-extinct, what's the plan for reproduction? Do you need like a genetically diverse population? Are you going to mate them with each other? Like what's the next stage?
So this is like, after you solve all of these impossible problems, now here's another set of impossible problems to deal with. Yeah, I think, if you imagine the world where we've created an Arctic adapted elephant, and we can do that once, we could probably do that several times. And what you would want to do is use different Asian elephants as the background, because then you have all of the genetic diversity that already exists in Asian elephants, and you've only modified the genes that were already fixed where there was no diversity in mammoths, right? So we've only changed these Asian elephants in ways that make genes that all mammoths had look like all of the rest of the mammoths. So it's not a place where you would really worry about inbreeding. Of course, if you just made one clone and then cloned that clone and bred them together, then you would have the problems of inbreeding that you might anticipate if you have an N of 1 for your whole population.
And people are already using this kind of tool for conservation purposes. Revive and Restore, this group Beth mentioned earlier, whose website and whose work I think is really fascinating. They've cloned a black-footed ferret from...
And Przeworski's horse.
Oh, and the horse, but the ferret was done intentionally from a cell line that's like 20 years old because it had different DNA than the existing living black-footed ferrets. So they want to breed it in to add some of the diversity that got lost as these things went through a near extinction bottleneck, to add it back to the species. That's stuff that's already happening on a very low, it's just starting on a very low level, but it's something you can do and should do.
We had a couple of questions that were about the idea of creating a genetic bank of DNA of species that we might clone later. These exist, and me and Colin were taken to one as part of research for Jurassic World. It's like a big cryogenic room with like a whole mix of like coolers. There's a whole cooler that's just like ferrets. It's like subspecies of ferrets.
Where did you go? Was it the one in San Diego at the San Diego?
It was in Washington. It was so it must have been at the is that the Smithsonian, the Science Museum, or yeah. So that was underneath the Smithsonian. There is a genetic bank, and many of them exist all over. I have time for one more question. It seems like, guys, crowd, I feel like we've kind of gotten to a lot of these questions. We've sort of circled up. Somebody particularly asked about the black rhino. How is the black rhino doing? Do you guys know anything about the status of that species?
Yeah, so it's the northern black rhinoceros. It's a subspecies, although these lines are tricky. The San Diego Zoo has a project going on with collaborators in various places. They're down to 2 living northern black rhinos. They're both female, they're both old, they're both too old to carry kids. They are working to try to recreate black rhino sperm and eggs from cell lines. They're working to try to make black rhino embryos that they can put into the southern. No, actually, it's not the black rhino. I'm thinking about the white rhinoceros.
White rhino, yeah.
Yeah. So, but all this is still true. It's just about the white rhino. They're working to make white rhino embryos, northern white rhino embryos they can put into southern white rhino females to try to get things done. It's the San Diego Zoo. Look up white rhino, San Diego Zoo, de-extinction. There's a lot of really interesting stuff going on there.
Yeah. Do you feel like there is a hierarchy of priority when it comes to our obligation to de-extinct species? Do you feel like there is some sort of moral good in de-extincting a species that other humans drove to extinction? Do you feel like that should be part of our consideration?
That's such an interesting question. There is this visceral sense of justice. You know, we butchered the Steller sea cow. Europeans took it from living to extinct in 18 years for no good reason. We butchered the passenger pigeon. There's a temptation to say we owe them a duty now that we can to bring them back, but that gets really tricky. Do you owe duties to extinct things? And you're not bringing back the ones you killed. You're bringing back sort of facsimiles of them, and how much human participation is necessary. In many cases, it's still very controversial about the role humans play, as with mammoths, for example. It's a really tempting argument. Ultimately, I don't find it a convincing argument, but I find it a really appealing argument.
Yeah. Yeah. Well, that's all we have time for, everybody. Thank you so much for your questions. And thank you so much to Beth and Hank for this fascinating conversation.
Thank you.
Our pleasure.
Excellent moderating. It's been fun.
Was fun. And Hank, you left me less confused this time around, but with no fewer provocative questions. And I think, Beth, despite my spoiler alerts, I think you've also left us with a lot of good science understanding, but a lot of good science questions as well. And I would imagine that if we have you back later, that as the science continues to evolve and you make new progress and new discoveries, that there will be more to discuss on this. So expect an invitation in your email box a couple of years from now so we can get an update from both of you. So
I want to thank everyone who asked a question. We had over 70 of them today. We did not have time to get to all of them. Emily, that was a fantastic job of moderating. Thank you so much. And I hope you'll consider doing it again for us. I also want to thank everybody who joined at the supporter level for this event. We could not do these events without the folks who donate. And Courtney Sloan, Sachi Gerben, Jeff Fishman, and Amechi Ukabi for technically producing and doing all the background work around this event. Join us. We're going to be off next week, but the week after we will be back with
Insect Wars, the barbaric way of bugs. Did I get that right?
I think you nailed it.
Okay. All right. Well, good.
This morning.
Good. Fantastic. Well, we thank you for being here, and we look forward to seeing you in 2 weeks. Thank you very much.
Thanks, everybody. Talk soon.